Good afternoon, ladies and gentlemen. I hope this is not going to fall on my head because it's not tight enough. So, thank you for the opportunity to speak here. I would like to introduce you to the world of chemical robotics and how we can use it for cancer treatment. Targeted drug delivery is a method how we are delivering the drug directly to the site where it should act. This can be, for example, a tumor. By delivering the drug directly to the site, we are evading healthy tissues and thus minimizing side effects to the body. There are already nanoparticulate systems on the market, for example Doxil, which is a doxorubicin formulation meant for cancer treatment. Another type of targeted drug delivery devices might be drug polymer conjugates. Both of these systems are accumulating in the tumor passively, which means thanks to their size and special construction of a tumor tissue. In laboratory of chemical robotics, we are developing nanoparticles which are smart and are capable of doing so many interesting things. The most important part is that they are carrying active payload in their core, which can be drug or prodrug. Important part of these nanoparticles is that they can find and bind the target, which can be the tumor cell, and this is done by adding moiety like an antibody to the nanoparticle surface. We can release the drug on demand by applying radiofrequency field on these nanoparticles if there are magnetic nanoparticles in their core. And last but not least, stealth surface coverage of such nanoparticles help us to prolong their circulation in the bloodstream, because these polymers we can use for stealth coverage repel plasmatic proteins, which would otherwise induce removal of such nanoparticles. In my last studies, I've been focusing on these two parts, which are the antibody binding and stealth covering of such nanoparticles. I published a paper on the topic where I'm showing that when we modify nanoparticles with monoclonal antibodies, we can increase their accumulation in the tumor like 10-fold times, and the retention in the tumor tissue is significantly prolonged when we compare it to the nanoparticles without the antibody. So we decided to use this surface modification to already a functional nanoparticle, and for this case we chose the liposome. Liposome is phospholipid vesicle, which consists of phospholipid bilayer, which is lipophilic, and hydrophilic lumen. This allows us to encapsulate both hydrophilic and lipophilic substances into such particles. Our liposomes consist of 3 types of phospholipids. One of them contains polyethylene glycol chains, which provide stealth surface coverage, and we also have reactive phospholipids with maleic groups on the surface, and these groups allow us to couple antibody. Size is adjusted by extrusion, and here you can see that our liposomes are around 200 nanometers in diameter. The next step is coupling of an antibody fragment to the nanoparticle surface. Antibodies are natural glycoproteins, and they are responsible for immune reactions of our system. In the first step, we are digesting antibody, and that's because we need to get rid of this part of the antibody, which would otherwise induce removal of our nanoparticles. Next step is purification and reduction. That's how we produce FAB fragments, which are responsible for binding an antigen. These fragments have free sulfhydryl groups, and that's how we can couple them to our liposomes. This is the step which we are now optimizing to produce high yield of FAB fragments. When we obtain these fragments, we will just couple them to the liposomes, and these liposomes can be intravenously administered. While being carried with bloodstream, when they find a tumor which expresses certain antigen, for which we have an antibody on the liposomes, these nanoparticles will be captured in the tumor. When we compare our liposomes with antibody to liposomes which are already on the market, our liposomes will be retained in the tumor tissue thanks to the antibodies for much longer time, and therefore will improve the treatment and the drug concentration in the tumor tissue. In the laboratory of chemical robotics, we're also working on related topics which would, for example, help us to release the drug on demand. This is preparation of magnetoliposomes. Another part, which is also very important, is improving the solubility of poorly soluble drugs. When we adsorb poorly soluble drug on the surface of mesoporous silica nanoparticle, and then we cap this nanoparticle with a liposome, the loading capacity of such particle is significantly increased. I would like to thank my colleagues for the cooperation and you for your attention.
Thank you very much for the lecture.
Thank you very much. I did not expect this.
And it's time for questions.
No questions?
So I would like...
There is one question?
Yeah, nice.
Cell line. Well, my antibody I'm using is IgG-M75, which is specific for carbonic anhydrase 9, and therefore we are testing on HT29 cell line, which is expressing carbonic anhydrase 9. And as a control, we are using another cell lines like DLD1. Both of them are colorectal carcinoma cell lines.
So, yeah?
What do you think about positive immune response in body?
Thank you. That's why we are removing Fc fragments of our antibodies. Otherwise, macrophages which have receptors for Fc fragments would recognize our nanoparticles. Always there is some immune response, but when the nanoparticle surface is stealth, the immune response is lowered, and also when there is only the Fab fragment of an antibody, we can say that there is a prolonged circulation, and these nanoparticles are able to circulate at least like 1 or 2 hours, which would not happen if the surface is not stealth. Such nanoparticles are removed in 2 to 5 minutes. So the immune response is always there. You just have to modulate it and decrease it.
The last question.
If I understand it well, your liposomes are not the only liposomes on the market with antibodies.
No. No. On the market there are already liposomes, but without antibodies. So liposomes with antibodies are not already on the market.
Okay.
We're just working on them.
Thank you very much once more.
Thank you very much.